Our culture system captures the phenotypic plasticity of lung squamous cell carcinoma cells in response to tumor stroma interaction, and how these interactions regulate tumor morphology during lung squamous cell carcinoma progression. The main advantage of our system is its ability to model lung squamous cell biology in a 3D context while preserving the plasticity of the malignant cells.
This 3D co-culture provides a unique system to investigate tumor stromal cell interactions and could be adapted to monitor responses of lung squamous cell carcinoma cells and cancer-associated fibroblasts to drug treatment.
To begin, thaw vials of basement membrane matrix in a 4°C refrigerator overnight. Cool down 2 milliliter plastic pipettes and tips at -20°C overnight. The next day, warm the reagents used to dissociate TUM622 cells, Heapy's buffer, trypsin EDTA, and trypsin neutralization buffer in a 37°C water bath. Take the thawed basement membrane matrix out of the refrigerator and put the vial on ice. Cool down the tissue culture plates on a metal platform cooler placed on ice. Place centrifuge tubes on a metal cooling rack on ice. Calculate the amount of cells needed based on the number of wells and the concentration of cells in each well to be prepared. Transfer 2M622 cell suspension into a cooled centrifuge tube and spin down at 300 times G in a hanging bucket centrifuge at 4°C for 5 minutes. With an